next speaker is Matt Guilliams. Matt is the plant systematicist and herbarium curator at the Santa Barbara Botanic Garden. Prior to working at the garden, Matt was a graduate student in the Department of Integrative Biology at UC Berkeley. At the garden, Matt works with the biodiversity team toward the study and conservation of California's remarkable flora, with a geographic emphasis on the Central Coast and Channel Islands. The title of Matt's talk is Looking for One Thing and Finding Another, Phylogenomic Study of Eriodictyon capitatum Resolves the Placement of the Enigmatic Namaceae. Thank you, Matt.
Hey, everyone. How's it going? Good, yeah. Thanks, Len, and thanks to Northern California botanists for having me here, and to you all for persisting through an awesome day of talks. Jim, awesome talk. Today I'm going to talk about a project that we've been working on at the Santa Barbara Botanic Garden, focusing on the genus Eriodictyon. Just a little bit of forewarning, it's a little bit late. There's going to be 2 opportunities for audience participation. Both will require your cell phone, so if you have your cell phone in your pocket and you want to participate, maybe you can get it out now. So we're going to be talking about the Namaceae, and this is what might be for some of you a new change that came out in the Jepson Manual and the eFlora a couple years ago. But it really harkens back to a phylogenetic study that was done by Ferguson in the late 1990s. And what she recovered in looking at the hydrophalaceae was that the hydrophalaceae form 2 main clades. In their analysis, these clades are sister with strong support, but we have one clade here, up here, that she's calling clade 1, that corresponds with what we think of the hydrophalaceae as today, which is Phacelia, Philistoma, and others. And then she recovered a clade 2, which contains the genera Nama, Eriodictyon, Wigandia, then Turricula. So this was our first hint that there was something going on. And it turns out that the hydrophalaceae and the Namaceae are not each other's closest relatives. So this is a tree from 2016, a little bit later, that shows the relationships in the Boraginales. This is the order that's named after the Borage family, which is the type family for this order. But this is where Namas and the Namaceae are situated, in with groups like Amsynchia and Philisma and Cordia, which doesn't occur here. How do you tell the Namaceae from the hydrophalaceae? Well, it's actually pretty darn easy. This is the entire protologue for the family description Namaceae. It's just like a line and a half, and it's pretty straightforward. So it's if you have 2 styles emerging from the top of the ovary, then you have Namaceae, and so you can see that. I think you can see that. This angle is so oblique. So you can see that angle there on Eriodictyon. You can see 2 styles there. Same thing here with Nama. But in that other clade, Phacelia being the example here, you can see that there's a single style emerging from the top of the ovary that has 2 branches at the apex. So that's it. It's literally that easy, telling hydrophalaceae from Namaceae. When we reduce the Namaceae down, when we partition it out into its own family, it's actually a pretty simple and digestible group of taxa. Only 3 genera, Eriodictyon being the focus of the talk today. So 11 species in Eriodictyon, Western North America. Wigandia, some of you may know from it's being planted around yards. But this is really a Central and Northern South America group of shrubs, 7 species. And then the hyperdiverse, largely annual Nama, 55 species, largely North America, but with a large intercontinental disjunction to South America, which is pretty neat. But if you're like me, you want to be able to key these things. And so this is how it keys out when you start out at the family level to the key to genus in the Jepson eFlora. And it makes some sense. You start out with woody plants here at the first lead, shrubs and small trees. This next, going with the second lead there, you have Nama partitioned out as annuals, but then we have this whole group of what I'm calling exceptional cases, and they're weird. They're just plants with different life histories that have made them difficult to place. And so this is former Turricula perrii, now in Eriodictyon. Eriodictyon labii, formerly in Nama, and then we have rothrockii, which is really what we'll talk about at the very end of this. So these are the exceptional cases. So here's your first opportunity for some audience participation. I should say, this is almost not safe for work. So if a lateral view of a butt cheek is going to bother you, then don't scan that QR code. But this is Eriodictyon perrii here, a picture of it. It's a beautiful plant, highly glandular. And this is one that causes, not to be rivaled contact photodermatitis. And so if you look at this person's experience with it, hiking the PCT, you'll have a lot of respect for that particular plant, what it can do to you. I'm not hearing any gasps yet, but it's pretty terrible. This one here, this is former Nama labii, Eriodictyon labii, and bizarre because it's a perennial herb. So if you think about Eriodictyons, you're thinking of rhizomatous perennial shrubs, long-lived, and this is a perennial herb. You can get a sense for what it looks like here. This might be 30 or 40 centimeters tall and with rhizomes underneath. And then rothrockii, similarly a perennial herb, but with these really, really neat kind of impressed veined leaves and this kind of pom-pom of flowers. So really a pretty cool looking plant. Okay, so we started out looking at Eriodictyon here at the garden. If you know Eriodictyon, you largely know it from probably one or more of the very common, widely distributed shrubs in this genus. So this is Eriodictyon californicum, really the only one north of San Luis Obispo County that's widespread. And it's really typical of Eriodictyons in having these narrowly elliptical to narrowly lanceolate leaves, usually pretty glabrous, a panicle of flowers, and these nice funnel-form corollas that can be lavender or white color. Down in my neck of the woods, we have that plant is largely ecologically replaced by Eriodictyon crassifolium, which has a similar shaped leaf and a similar funnel-form corolla, but the whole plant is hairy. So the leaves are hairy, the inflorescence axes are hairy, and the corollas are hairy. Trichocalyx is more of a high elevation species in the transverse and peninsular ranges, getting down to getting down to lower elevation in Baja California. That's a story for another time, but readily identifiable as an Eriodictyon, white-flowered or violet-flowered, depending upon the subspecies. And then Angustifolium, this is where there's a little bit of evolutionary innovation here with the leaves being linear. And this is a plant that occurs in the desert southwest and then disjunctly to northern Baja California. Now, we got started working on Eriodictyon because the Fish and Wildlife Service was hoping for some information to help them manage the species. Two different federally listed species. This is one, Eriodictyon capitatum. This is a CMPS list 1B1, federal endangered and state rare. And this is such an awesome plant. It really just grows right around the Lompoc area and in the western San Ynez Mountains. It's got these beautiful linear leaves, gummy, aromatic, resinous. And then very unusual for Eriodictyon is it has a capitate inflorescence. So there's a gathering of flowers, just imagine the internodes very short and all the flowers clustered together. So that's a great plant. That's the one that we did some population genomics on, along with this one, Eriodictyon altissimum. This is the Indian knob mountain balm that's known only from San Luis Obispo County, around Los Osos, and the city of San Luis Obispo. Similar to capitatum in that it has these beautiful, gummy, wonderful, smelly, narrow leaves, linear leaves. But instead of the capitate inflorescence, has got kind of an open panicle. And this led to, in the prologue, when Phil Wells described this plant in 1962, he speculated at the end, he wasn't real firm about this, but he just speculated that perhaps this combination that he saw in Eriodictyon altissimum and that I just described came from the ancient interaction or hybridization between Eriodictyon, capitatum over there with the linear leaves, and then something like californicum that has these open panicle inflorescences. But there was a big problem with this particular idea is that neither of those plants occur there today. So it's stranger things have happened, but it was a problem. So we needed some other source of data other than morphology, and our genomic data presented a nice opportunity for looking at this. Okay, so we've been looking at the genetics of Eriodictyon for maybe 8 or 9 years now, and we're mostly looking at population genomics of these two rare plants. But this was also a nice opportunity to look, and what I'll be talking about today is the phylogenetics of the genus. and with particular emphasis on trying to understand whether or not the phylogenetic data and analyses support the circumscription of these two rare plants. We can also evaluate Wells's hybrid origin idea for Eriodictyon altissimum. And then really the punchline for today is to confirm the placement of those three exceptional taxa. 5 minutes. Wow. Okay. Okay, we're going to go ahead and skip the methods. real happy to talk anytime about this. I should have given a little trigger warning. Some people don't like looking at phylogenetic trees. I love them. These were, it was really a successful project for thinking about evolution of Eriodictyon. A couple things here. We have really good support throughout the tree. So we have about 2.2 million base pairs worth of data that we analyzed for this data set. And it provided a lot of resolution. I'm really only showing a couple of nodes here that don't have maximum statistical support. But otherwise, the tree has a lot of good support. Most of the taxa where we sampled multiple individuals per taxon were monophyletic with really strong support. And that includes our two friends, capitatum and altissimum. So that's awesome news for the resource agencies that we have a lot of molecular support for the circumscription of those plants. But check out where Californicum occurs. It's a totally separate part of the tree. We have no evidence at all that Californicum interacted with capitatum to spawn altissimum. So no support for that idea. Eriodictyon perii, this is poodle dog bush, Turricula perii. This is, in every single one of our trees was recovered in a clade with Eriodictyon, but this node here with really poor support lets us know that this clade of Turricula or Eriodictyon perii bounces around inside of this tree. So from that, we would infer this was a rapid diversification. Even with 2.2 million base pairs of data, we can't place it sufficiently, but including it in Eriodictyon is a good idea. Eriodictyon labii, this is a plant from up in this part of the world, the perennial herb, falls out with Eriodictyon with very strong support. And here's our buddy here, Namaceae. Namaceae is on recovered with 100% or 100 bootstrap, maximum likelihood bootstrap support on a very long branch, sister to a long branch leading to Eriodictyon. So that is what I want to say about that. I'm going to move through biogeography super fast. So the take-home messages here are that this molecular approach worked really well for Eriodictyon, worked great for population genomics. We have a well-supported altissimum and capitatum clades. They're sister to each other, have no interaction with Californicum. We have good placement of the exceptional taxa, and that leads to this part of the talk, which is really the punchline here, is that Namaceae needs a new name. It's not a Nama. Nama that we sampled are over here. So some well-supported phylogenetic structure shows that it's sister on a very long branch to Eriodictyon. And this is the second part of the audience participation. There is a very lively discussion in the literature in the last 3 or 4 years about who gets access, who has the agency to do things like name things in science. And with that in mind, I thought it might be kind of fun to crowdsource the naming of this plant. So I have no druthers. It doesn't matter all that much to me. I don't have a dog in the hunt, but maybe you've got a great name for it. The other alternative that would work equally well is to lump it in with Eriodictyon, but my preference at this point is, because it is on a long branch and it is so morphologically unique, to give it a new genus name. So if you'd like to, you can scan this QR code. It'll take you to a Google poll, and you can provide as many examples, well, up to 3, I think is what I put on there. But feel free to suggest some ideas, and maybe your name will win. So with that, I'd like to just acknowledge folks for their help with sampling for this, folks who are listed there that I won't read out just for time. Especially would like to thank U.S. Fish and Wildlife Service for supporting our permit to work with this rare plant. A number of people in this room helped with samples and for funding U.S. Fish and Wildlife Service. Thank you. It looks like there might be time for one or two questions. Awesome. Okay, thanks for your time.
Thanks, Matt.